Okay, so welcome everyone. This is a reboot of the Sheep Reproduction Strategic Partnership webinar series, where generally we'll be working on a quarterly basis and just bringing some topics of interest and of use to sheep producers related to sheep reproduction to a wider audience on, again, as I said, on a quarterly basis. So today our focus of our webinar is all about the ram side of the sheep reproduction process, and under the theme of Let's Talk Rams. We've got two speakers today from the University of Sydney, but before we start, we'll just go through a quick run through. So again, thanks for joining the webinar. Anyone that's joining, make sure you've got yourself muted and your video is off. If you have any issues, try phoning in on the number on the screen using the meeting ID and the passwords. And don't forget to ask a question. We'll just do that during the chat function. So if you have any questions that come up during the two presentations, use the chat function. I'll keep an eye on that and I'll direct the questions to the speakers at the end of their talks. So the Sheep Reproduction Strategic Partnership, it's all about sustainably increasing sheep weaning rates through increased survivability and decreased mortality. So we work throughout the sheep breeding cycle, from joining all the way through to weaning, and then in that sort of pre-joining period as well. And what we try and do is to promote best practice through the management cycle to make sure that we're getting best results out of our breeding ewes, out of our rams, best results from lambing, and also managing that weaning process, and also that really important pre-joining process to ensure our animals are in the right condition at joining. So we promote best management practice, and through the partnership, try and fund projects where there are gaps in the research to make sure we can provide useful, practical information for producers to implement on farm. Our SRSP website, we're trying to develop this into a one-stop shop for sheep reproduction research information. It's just gone through a revamp, so there's some new information on there. So I encourage everyone to have a look. Have a browse through there, see what's on there, and if there's anything that you would like more information on, just get in touch with myself as the program coordinator, and we can work to continue the work to make sure this is a really used and informative and useful resource for industry. So that's enough from me. The first of our speakers today is Professor Simon de Graaf from the University of Sydney. He's going to talk to us about sex semen and its role in sheep production systems. So while Simon gets himself ready to present, I'll give a little bit of an introduction. He's currently Professor of Animal Reproduction at the University of Sydney and Secretary General of the International Congress on Animal Reproduction. He's Director of the Research Company and also host and producer of Repro Radio podcasts. He's recognised as a worldwide expert in sheep reproduction, artificial breeding and semen assessment. And so I look forward to hearing I'll stop sharing. Look forward to hearing all about sex semen in Australian sheep systems. Thanks, Simon.
Thanks very much, Sue, for that wonderful introduction. Lovely to be with you all here today. So as promised, I will be speaking primarily about sex semen. I probably won't talk too much about how you would use it in your particular production systems. You can certainly ask me towards the end if there's something that you would like me to cover specifically. But broadly speaking, we'll be talking about the history of the sperm sexing process, how it works. It's probably one of my most frequently asked questions that I receive on a day-to-day basis is, so how does that sexing process actually work? So we'll cover that, and how it was developed, and then we'll talk about the current status in the sheep industry, and I will try and remember there to touch on how you might want to use it, and then we can chat as well about what sort of research is needed to try and take things to the next level with sex semen in sheep. So I always like to start to be honest with you, I like starting most talks that I give with some kind of rocketry, because I think it's really cool. I wanted to be a flight engineer, aeronautical engineer, a pilot. I ended up in agriculture, no regrets, but I do kind of come back to this world whenever I can. So this talk is no different. You might wonder what it is that links sex semen to the development of nuclear weapons, the development of intercontinental ballistic missiles, and you would perhaps be surprised to discover that it was all invented in the same laboratory. So up there in the top right of your screen, you can see Lawrence Livermore National Laboratory. It's a very, very famous laboratory in the US where these weapon systems were developed. And around about the 80s, well, late 70s, early 80s, there was a research project going on where, like, obviously there's a lot of nuclear development going in there. One of the research projects was around trying to figure out what sort of impacts radiation had on DNA, and what sort of effects radiation had on the DNA in sperm. But there was not the means to assess that appropriately. There are these machines called flow cytometers, which allow you to analyze certain characteristics of individual cells. But sperm cells are a little bit funny. They're a little bit different to the normal round cell. They behave differently. And so they had to develop a special type of flow cytometer in order to measure whether or not the DNA in mouse sperm was being damaged by nuclear radiation. And some particularly smart cookie happened to be around at that time doing a bit of sabbatical from the mammalian reproduction and livestock systems world, happened to be there in Lawrence Livermore Labs and saw what was going on and saw the potential application to what had long been wanting to be developed, was this idea of being able to separate male and female sperm. And so it was from that in conjunction with the USDA, they developed what was initially referred to as the Beltsville sperm sexing technology. And so the way this works is that there is a modified flow cytometer. So this is just a machine that allows you to deliver a single cell to a fixed point at a specific time. So imagine you attach a little tube of diluted sperm to a machine, and it sucks that sperm up, and then it shoots it out of a little nozzle in a big long line, just a single stream, one by one after each other, and it shoots them out there at about 80 kilometers an hour. And there's a laser that interrogates each of the individual sperm, and each of these sperm is encased in a single droplet of fluid. When that laser interrogates the sperm, it's looking to see how fluorescent that sperm is. Now, the sperm are only fluorescent because they've had a special dye added to them called Herxt 33342. And that dye binds with the DNA. And if you are a sperm that has an X chromosome, so that will, if it fertilizes the egg, will end up in a female offspring, then you have slightly more DNA inside of you than if you are a sperm with a Y chromosome, because the X chromosome is bigger. So that means that there's more fluorescence that can be picked up by the machine, and then the machine can tell the droplet to be charged with a positive charge if you're an X-bearing sperm, or a negative charge if you're a Y-bearing sperm. And then when you pass through two electrostatic gates, you can be drafted. You just push one way or another into a tube. So that's roughly speaking how that technology works. And whilst there have been a lot of updates over the years, it's still fundamentally the same. Those patents are still essentially the same. So it took a while to go from those initial discoveries through to some live births. The first live births following AI were in rabbits. In 1989, this work was published in Biology of Reproduction. And from there, it was very rapidly pushed out into other species. So it went from rabbits to pigs, and then to cattle, sheep, humans, horses, and then onwards from there. Now, to begin with, the process was so laboriously slow that you could only use sexed sperm with additional technologies. So like something called ICSI, where you can just put a single sperm into an egg to get fertilization, or IVF, where you have some eggs in a dish and you put some sperm in there. Artificial insemination, where you need quite a few more sperm cells, it just weren't at that point yet. The sorters weren't fast enough and the sperm wasn't good enough either. Now, the main commercial focus, of course, has been on the cattle side of things. It was initially commercialised in 1999 with Cogent in the UK, then XY Inc. and at some point in the late 2000s, I think it was around 2007, 2008, one of their licensees, ST Genetics, bought out XY, and they are now the sole licensee of this technology from the USDA. Of course, we're interested in sheep, so we will talk a little bit about how we've gone from 1995, where we had the first live births here. Well, it's actually one live birth. Larry the lamb was born here at the University of Sydney, just in that building there, out my window, from a intracytoplasmic sperm injection to today in 2023, when there have been really tens of thousands of births of pre-sexed lambs. So you can see in this particular slide, there's a whole lot of, this is the first time we did whatever, and essentially what you're seeing is development from more intensive reproductive technologies to less intensive reproductive technologies in order to obtain a pregnancy. And you're also moving from having fresh semen to having frozen semen. We even had a period where we had frozen semen that was thawed out, sex sorted, and then refrozen, and then thawed out again. And then we finished a lot of this research work, and it was moved into the commercial space in the late 2010s, and it was really released to market in Australia and around about, it was 2020 realistically, but from around about 2018 internationally it could be bought. So what's the current status? Well, if there's anybody listening that has done AI, you'll know that you can inseminate fresh semen or you can inseminate frozen semen. Frozen semen is certainly easier, particularly if the animal is not right next to you. So with sex sperm, there's been some issues with developing the sexed frozen product in the commercial setting. We got some great results back when we had it in the research environment, but in the commercial environment, there have been some issues with getting a robust frozen product. So for ram semen, for sex ram semen, most of it is what they refer to as a liquid product. So what this means is it's collected, it's sexed, and then it is overnighted to wherever you are, and it's not frozen, and it's AI'd directly as still a fresh sample. Now, if you're curious about how well it works very well in terms of its sex ratio. So you should get 90% plus of the sex that you desire. And the in vitro quality of the sperm is generally, it's pretty good. It looks quite good when it gets to the property. As to the fertility, it has been a little bit variable. You know, there are people that really have had wonderful experiences, and then there are some that have had experiences which are not quite so wonderful. And there have been some notable cataclysmic sex programs that just, they haven't gone so well. Overseas, particularly in South Africa, they've had some really great results. And then here there's been, as I said, almost exclusively fresh liquid, but there have been just a handful of sex frozen programs, and there have been examples where that has worked well, but more often than not it hasn't been great. So we counsel caution with the sex frozen ram product. And the questions always arise, why is there this variation with AI results? Why is one person getting a great result when somebody else is not getting such a great result? And for sex semen, part of the issue is there's substantial variation in the synchronization programs which are used with sexed sperm. And they also are what I would classify as fairly high risk AI programs, because as a general rule, they're only using a single sire. Now, just normally, if you take sexing out of the equation, use of a single sire is a little bit dangerous, because if you have some kind of a fertility issue with that one male, then you are going to, doesn't matter how well the animals are cycling, you're going to have a poor AI result. And that just happens sometimes. That's male factor infertility or male factor subfertility. Now, what happens when you drop AI doses down? Because the AI dose which is used is not the standard that you get with conventional semen of being 25 million motile. It's down around the 4 to 6 million total cells. Now, when you drop AI dose, even with conventional sperm, you'd get a more variable result. And some sires, they hold up well. You can see that in that graph there. As you are reducing the total number of sperm in an AI dose for each of the sires, some of them are totally fine. They're all good until very low numbers, whereas others, they fall away quite quickly. And I would suggest that perhaps what is occurring is that there are some AI sires which aren't well adapted to this low dose insemination. So it may be because they're undergoing low dose AI rather than the sexing process itself, which is causing that program to have a poor result. So what needs to be done? I'd say there's probably still some work to further refine the processing of sex ram semen, and SG Genetics are interested in further exploration of some of this. Within their bull product line, they have something called Sexed Ultra Plus, which is really the result of a lot of work over the years to improve the processing, the type of media which is used, and what have you, in order to boost that sperm quality and the chances of fertility. There needs to be further research in Australia to refine that AI timing and synchronization of fresh and frozen sex sperm, and probably a bit more work around sperm dose. So, is it okay to just use 6, or realistically, do we need to use 8? Do we need to use 10? And what do we do when it's fresh? What do we do when it's frozen? And if we have superovulated donors, do we need to use a couple of straws? What's the issue there? And those trials need to not be just somebody having a crack with no decent controls. You need to have controls in there with conventional semen, and comparing apples with apples is really important, because we know that it can work. This graph shows along the bottom number of motile sperm inseminated. The yellow bars are sexed semen. The blue bars are non-sexed semen, and the y-axis is pregnancy rate. And you can see that when there's 15 million motile sperm cells inseminated, you're getting the same results. When you drop down to 5 and 1 million motile sperm, we were actually getting better fertility with sex semen than we were with conventional semen, because we think that the sorters are able to pick out the best sperm cells and get rid of the ones that are less likely to be fertile. So we know it's possible to have a really robust sex frozen product. There's just probably a little bit more work there needed. So to wrap up, what are the lessons from this kind of sex semen developmental pathline? I'd say innovation can come from unlikely places, and R&D timelines are quite long. From the first development of that flow cytometer in the early 80s to sexed ram semen being released in Australia in 2020, it's a pretty long period. It's about as long as I've been alive. It's also worth noting that blue sky research can in fact pay off. Sometimes it's worth drawing a long bow and seeing what can happen rather than saying, all right, we want to see some sort of practical outcome in the next 6 to 12 months. You know, it can really stifle innovation. Some of the practical take-homes, a reminder for all the reproductive biologists out there that sheep are not cows. I think it's probably worth all ag consultants to remember that as well. They're not just small cows. And when you're thinking about risk management in an artificial breeding setting, remember to try not to rely on just one sire, have multiple sires. And other than that, we really need some research at scale to try and solve a few of these issues around AI time and the protocols necessary in order to boost our confidence that we're going to be able to get good fertility with all of the sexed products in all of the settings. So for more information about what I've very briefly covered here today, including some of the stuff about when you would actually use sexed semen, you can take a look at this blog post that I wrote last year. There's a little QR code that you can just go straight to that site if you pop your phone up there now, or you can go to that web link. There's also a podcast episode that we did with the manager of the sperm sexing lab at ST Genetics down in Camperdown in Victoria. And there's some information there right now about if you're thinking about doing a sexed ram semen program. Step one is talk to your breeding company, and they'll probably cover all of that with Total Livestock Genetics down in Camperdown. So with that, thanks to my team over the years that has worked on all of this over several decades, and all the people that came before me and afterwards. It really was a labour of love to develop that technology and get it released to the industry. And with that, I'd say thank you.
That's great, Simon. Thanks for that really good overview of sex semen. I have to definitely agree with two of your points. Yes, definitely agree there is a need for blue sky research and a balanced portfolio to make sure we have something coming through the pipeline. And it's not just reproduction biologists that need to remember that sheep are not just small cows. I think that goes for everyone who works in animal science, that sheep are not just small cows.
Absolutely.
So we've got a question first. So Catherine thanks you for your talk. And she's wanting to know, is there much industry update of sex semen in sheep?
Industry uptake.
Yes.
Yeah, it's hard to get numbers, exact numbers on any kind of AI programs, but it seems to be growing and growing enough that ST Genetics say that it's no longer just something which is happening every so often. It's something that they need to factor into their bookings in a major way. Mm-hmm. So they would be getting probably a lot of goats as well, a lot of dairy goat work, and collectively, I think the last time I was speaking to Neil, it's still small fry in comparison to the amount of time that they have their sorters operating for cattle, for dairy cattle. But I'd say around about 5% of machine time would be small ruminants. So and that it's growing, and they're really excited about that. So yeah, it seems to be something that is being used, which is nice, and used in a variety of different ways too, which is a handball question to Sue to ask me about. Simon, what ways would people use?
I'll do that one in a second. But the follow-up question from Catherine was, do you think that the amount of industry uptake would change if sexed frozen semen was available?
Oh yeah, like 100,000%. I know in particular in WA, there's an awful lot of people that have shown an awful lot of interest, and it's really difficult. Shipping across the Nullarbor it's hard. To be really fast and really reliable, that's tricky. So I mean, like the way that the process works is that you've got to get your ram there to Camperdown in Victoria, collect, do the sexing, and then get it back by the next day. And so, yeah, you're really limited by the fact that it's fresh. So it's the big reason why. I mean, my PhD was all about improving the frozen quality of sexed sperm, because we knew that would be the thing that would really drive uptake. So I would imagine it would explode in use if that was the case.
I think you're exactly right. Just the logistics of Australia, and especially and getting it to a capital city and getting it out to regional areas is even more difficult. So okay, here's the handball question coming back at you. Where do you see its most benefit to come from for sheep producers, in the seed stock sector or in the commercial sector?
Look, I suppose that AI has always got more of an uptake in the seed stock sector. The dollars make a little bit more sense there, and the way that people use it is they use it in different ways. There are people that are, their main business is selling rams, and they want more rams, and so they're able to just inseminate Y-bearing sperm and end up with more rams to sell, or more rams to select from, for their sales or whatnot. But then there are other people that I speak to, and they're like, oh, look, we really want to work on this female line, and it'd really be handy if I had more females to select from in a drop, as we really want to develop this line. So there are people that will use it, X-bearing sperm. So yeah, it's different depending on different settings. There's probably situations where maybe the I guess the dollars would add up to potentially use it in a more commercial type operations than seed stock. I'm hoping to have an honours student next year to work up some of the economic cases around this and some other reproductive technologies. So
yeah, no, that'd be really, really good. So, using my chairperson's prerogative and my background interest in genetics, I was really interested in that sort of individual variation between size you talked about. And, is there any sort of correlation between the variation between size in the success and any other key production traits? So is there any indication that, high fleece weight or diameter or fatness or eye muscle depth, is any of that correlated with the success? So are we looking at maybe some genetic antagonisms that could be at work there?
Yeah, look, the answer is I don't know. The numbers are far too small that have been done to be able to draw any of those kinds of conclusions. But with enough scale and with enough knowledge as well of what's being inseminated when, you would be able to start doing that kind of thing. I suppose the thing to remember with a lot of the sheep work over the years is that when they're done in some of the research settings, there's not a lot known about the rams really, other than they're producing good semen and they're pretty reliable. But as there are opportunities to use animals in, I guess, trial work that is done within an industry setting, where we know more about the genetics of those animals, we would be able to start drawing some of those conclusions, because it's not an unreasonable thing to think would be potentially occurring.
That's right. And you want to hope there weren't any serious antagonisms with some of the key traits we select for in breeding programs. But I guess that's something we park until frozen semen is widely used and we can get all those extra animals measured and being used, I guess.
Absolutely.
All right, so we might just draw that to a close, and we'll sort of go into the second part of today's webinar,